welcome today's speaker, Joe Takahashi from UT Southwestern. He's the chair there of the Neuroscience Department and the Lloyd B. Sants Distinguished Chair in Neuroscience. And Joe received a PhD from the University of Oregon, working in the lab of Michael Menaker, was already interested in circadian rhythms, which the field to which he's devoted his career. He went from there to the NIH for a postdoc, and after that joined the faculty at Northwestern, where he made discoveries that I consider among the most impactful ever in the fields of both neurogenetics and behavioral neuroscience. First, in a paper in Science, described the genetic mapping of the mouse clock gene, which he and his colleagues did using an EMU mutagenesis screen, where they were able to show the chromosome location of a gene that affected both periodicity as well as the additional circadian phenotype of persistence of rhythm in the absence of external cues, and showed that this was clearly the same gene. And this was published in a paper in Science in 1994. And then a couple of years later, then demonstrated that, in fact, they'd identified this gene through the positional cloning of clock, which they then proved was playing this important role in circadian phenotype by rescuing the mutant phenotypes by use of a transgenic mouse that had a back containing the wild type clock. And, I think the impact scientifically and on medicine was really immediate, led to the subsequent years of work, which demonstrated the incredibly tight regulation of the circadian system with identification of several additional crucial genes that are part of this system, and then the demonstration, which Joe's lab played a leading role in, and then labs throughout the world joined in on, to show the importance of this system to virtually every aspect of mammalian biology and health, including the regulation of metabolism, the regulation of immunity, and really there's probably not a single sort of major human health disorder that in some way or other hasn't been shown that the system that this discovery led to the identification of plays an important role. I should say that it had a really personal impact on me in that I was still at a fairly early stage in my career in trying to do genetics of neuropsychiatric disorders. And I had spent a lot of the first part of my career figuring out how to do human genetics and hadn't spent a lot of time thinking about phenotypes. And I heard Joe give lecture on these discoveries fairly soon after the Cell papers appeared, when I was a faculty member at UCSF. And it really was like a light went on, to really demonstrate that, of course, the foundation of doing genetics of behavior in a complex organism is having an imaginative and rigorous way of both delineating and measuring behavioral phenotypes. And, and that, like I said, that really set, I think, both my course of, what I've done ever since then, and that of many others around me, influenced in quite the same way. Joe moved from Northwestern to UT Southwestern 2009 to take on the leadership of the Neuroscience Department, has done an incredible job there in recruiting scientists so that it's become a really amazing department. His list of honors and awards covers 2 full pages of CV. So I won't say much about them, but as probably most of you know, he was an HHMI investigator for more than a quarter century, as a member of National Academies of both Medicine and Science. And in the last few years, his lab has focused on a topic that probably many of us are concerned about, which is really trying to understand the aging process and longevity, and has really begun to show that the circadian clock may offer a way of really intervening in this system in a therapeutic way. And that's what he's going to talk about today. So please welcome Joe.
Thank you so much, Nelson, for that extremely nice introduction. Hope I can live up to that prelude. What I'd like to do first is maybe summarize a little history for you, which Nelson sort of told you about, and then move into what we're really focused on today, which is the role of circadian clocks in aging and longevity in mice. So as all of you know, we live on a planet with a 24-hour environmental cycle. We like to think of it as an energy cycle. If you were a plant, that would be obvious. You'd harvest energy in the day, do other things at night. But all living systems have evolved clocks to anticipate this energy cycle. And the common motif for these clocks is a negative feedback transcription translation cycle like this, which takes about 24 hours to complete. Now, I would say that the beginning of our molecular understanding of clocks began with this singular classic paper done over 50 years ago by Ron Konopka and Seymour Benzer at Caltech, where Ron, when he was a graduate student with Seymour, this is a photo much later, 2000, isolated or did a mutant screen in Drosophila for circadian rhythms, and he isolated 3 mutants, one that had no rhythm, one that had a short period clock, about 19 hours, and one that had a long period clock, 28 hours, okay? And they all mapped to the same locus, which they named period back then. And then it took many additional years to actually find the gene, because cloning genes was difficult back then from mutant phenotypes. And so this didn't happen until the mid-'80s. And that was done by Michael Rosbach and Jeff Hall at Brandeis and Michael Young at Rockefeller in 84, 85. But during this period, lots of us tried to isolate mammalian orthologs or homologs of the period gene through nucleotide sequence similarity. And we all failed. In fact, it wasn't accomplished until 1997 by a lab in Japan. And that's because at the nucleotide level, you can't recognize the period gene. You have to translate it into its amino acid sequence, and then you can finally recognize it, okay? So because of that inability for us to really go from flies to mammals, we decided to take a step backwards and do a phenotypic screen. And this was led by a really talented postdoc of mine, Martha Vita-Turna. We collaborated with Bill Dove at the University of Wisconsin-Madison, who was using ENU to mutagenize mice and screen for point mutations in an enzyme in the liver. And so we teamed up with Bill and his wife, Alex, and they did the screen and then shipped the mice off to us at Northwestern, and Martha then screened their behavior. And so our assay is to look at their spontaneous wheel running activity, plotted as actograms shown here. These are double plotted. And this is a pretty long record initially when we were screening these mice. They're on a light cycle shown here for 2 weeks, and then they go into darkness. And this is a record typical of a normal wild-type mouse. Okay, so it entrains, it runs at night. And then when it's in darkness, it has a short period. It wakes up a little earlier each day. This is clock time on the x-axis. And in this screen, Martha isolated this mouse. It can still synchronize to light, but when it goes into darkness, it has a 28-hour clock. It wakes up 4 hours later each day. And then after a couple weeks, it loses its circadian rhythm, and what's left is this episodic pattern of activity, which is not controlled by the circadian clock. Okay. Now, this mouse, as Nelson said, we were able to map to mouse chromosome 5. And during this period, I should just take an aside and say, there are incredible skeptics. They thought we were crazy when we did the screen. But then we were lucky, we isolated this mouse. But then when we had the mouse and we wanted to then find the gene, they said, oh, that's going to be really hard. You better let Eric Lander help you clone the gene. Now, Eric Lander did help us out because he was starting his mouse genetics genome center back then. So we used a lot of Eric's resources to eventually map at high resolution the mutation. But then other skeptics in my field said, well, you know, even if you find the gene, it's going to be CAM kinase 2. And it might not tell you anything about the clock. But the minute our paper came out with the product of the clock gene, which showed it was likely a transcriptional activator because it had a BHLH domain, a PAS domain shared with the original period protein, and then a glutamine-rich C-terminus, which is typical of transcriptional activators. The whole attitude changed. And so that was really one of the most wonderful periods of my career, I have to admit. And so clock, you know, was interesting because the period protein was very mysterious. We couldn't figure out what it was doing based on its amino acid sequence. And it only had this domain here called PAS, which was shared among 3 known proteins at that time: period, ARNT, and SIM, which turn out to all be BHLH PAS proteins. So many years later, at Southwestern, we are able to solve the crystal structure of the BHLH PAS-AB domains of this heterodynamic complex. And clock, of course, has a partner. It's called BMAL, which is also BHLH PAS-AB transcription factor. And these are the PAS domains, which we also find in the period protein. Okay, now they're very interesting. They're protein dimerization interfaces, but they also have a pocket. And in a number of members of this superfamily, such as HIF1 and HIF2, and the aryl hydrocarbon receptor, the PAS-B domain is actually a ligand binding domain, can bind small molecules. And it turned out to be an interesting protein. So during this period of 97 to about 2000, the whole field converged. And we initially showed that clock BMAL could transactivate the period 1 gene, which had just been cloned in mouse and human. And indeed, it could transactivate the fly gene at this time. And then a few years later, a second set of transcriptional repressors were shown to be part of this feedback loop, the cryptochrome proteins, which are also regulated by clock BMAL. And so these sets of genes are transcribed in the daytime. Proteins are translated. They dimerize, interact with each other and with other proteins, in particular casein kinase 1. Then they translocate into the nucleus in the evening, and then they repress their own transcription by direct physical interactions. So I'll show you in a moment. So this is what we call the core feedback loop. The clock gene network is actually much more complicated now. I'm not going to bother to show you all the additional loops. But what's important is that even though there are 3 loops, clock and BMAL actually regulate all 3 of those loops. So they are still the dominant master transcription factors that control the circadian gene network. And one of the major ways it signals emerge from this molecular clock is by regulation of many other target genes. So one of the surprises, I must have skipped it. No, I guess not. Okay. One of the surprises when we clone these genes is that their expression was ubiquitous. And so we and other labs showed that many tissues in the body actually have circadian rhythms. And this is just an example. We made a mouse that has luciferase fused to the period 2 gene to form a HER2 luciferase fusion protein, which is a very nice reporter of HER2 abundance. And with imaging, you can then measure single cell rhythms. So this is 2 examples of fibroblasts cycling for 18 days in this case. And these cells will cycle for as long as we can keep them alive in culture. We've gone 45 days easily, and the rhythms do not damp out. So at the cell autonomous level, this clock is incredibly robust. And so in the body, we now know that all of our major organ systems have clocks that are autonomous, and they're driven by this cell autonomous transcriptional feedback system. Okay, so this has led to a number of questions at the system level, which I'm not really going to talk about today, because initially we thought the clock, was really located in the brain and the hypothalamus. And so it was a surprise to see clocks everywhere. So what are these clocks doing? So just give you one example. My close colleague back at Northwestern, Javas, we noticed in the original clock mutant that they had a metabolic defect and they had a loss of glucose regulation. So eventually we made conditional knockouts of clock and of BMAL in pancreatic beta cells. And what we found is that loss of BMAL just in beta cells leads to a loss of glucose regulation, shown here relative to controls, and that this is really due to a deficiency in insulin production. And Bass has gone on to show it's really a deficiency in insulin secretion from pancreatic beta cells. So that's just one example of a new function back then for these circadian genes in peripheral tissues. Now, going back to this core feedback loop, over a decade ago, when next-gen sequencing became available and we could use ChIP-seq, we could then try to identify the targets of this transcriptional network using ChIP-seq technology. So here's just a browser view of BMAL occupancy at a known target gene called DBP, another transcription factor. Has 3 sites: promoter, intron 1, intron 2. And then these are samples taken at 6 times a day, every 4 hours. So 0 is dawn. So 0 to 12 is daytime, and then 12 is nighttime. And what you see is occupancy is high in the daytime, and then it becomes low at night. And then this is a BMAL knockout control for specificity. So the occupancy of BMAL is circadian or rhythmic. If we then look at the rest of that core transcriptional feedback loop, clock and BMAL have the same pattern, both temporally and spatially. And then the 4 repressors bind the same sites, but they come on at night during the repression phase between 12 and 20. And then a new feature came out of these experiments, and that was that Cry1 had an even delayed pattern. So it came on very late at night, peaked at the next morning, and then persisted into the next cycle. And so we now think of this as having 2 repressive phases: the classic repression phase in the late night, and then a delayed repressive phase that goes into the next cycle on the next day. when we looked at chromatin marks associated with Pol2 transcription, we found another surprise back then, and that was, for example, this classic promoter mark, histone 3 lysine 4 trimethylation, was highly dynamic. So for example, here at 0, you don't see the mark at all, okay? Then it peaks around 12 and then it goes away. This happens to over 5,000 sites at genes in the mouse liver. And so this is quite surprising because, you know, at the RNA level, we could detect maybe 3,000 RNAs that are cycling in the liver. But at the chromatin level, we can see that the effect of the clock is even more pervasive. Almost half the expressed genes have some kind of chromatin modulation that is cycling in the mouse liver. And it's pretty much coordinated. And so Nobu Koike, who did all these experiments, showed that in the mouse liver, there's this incredible stereotype transcriptional cycle that occurs every day. There's this activation phase where clock female occupy DNA, they recruit coactivators such as CBP, P300, and then you can see some activation marks like histone 3, lysine 9 acetylation peaking here. This is followed by a burst of transcription. You can see Pol2 occupancy peaking here, and then followed by the classic repression phase shown by the per genes shown here. And then we go into the next cycle where Cry1 is still bound to DNA for this late repressive phase, where clock and B-mail are actually bound to DNA, but there's no transcription because we think it's being repressed by cryptochrome 1 until it turns over, and then the cycle starts over again. So when we looked at, you know, what are the gene targets of this pathway, the top category, of course, is metabolism, which is not surprising for the mouse liver. So here's a chart of all metabolic pathways from KEGG. And overlaid in red are all the B-mal target genes. So, you know, it speaks for itself. The entire chart is covered. And so what I think it's telling us is that the clock is really regulating metabolism in a very direct cell autonomous manner. And it's impacting essentially every metabolic pathway in the cell. Now, since then, this has been done in a few other tissues, pancreas and some and brain tissue. And we actually see the same picture. It's not just in the liver where we see metabolism as a top go category. Okay. And so I think this is telling us something important about what the clock is doing. It's really regulating, fine-tuning metabolism and energy in a cell autonomous manner throughout the body. And maybe it makes sense. We used to think, you know, there's a master clock in the brain, and then it would signal to the liver. But that would be kind of an awkward, you know, way to control the transcription of all metabolic pathways in the liver to signal from the brain all the way down to the liver, mobilize a new transcriptional network to then turn it on and off each day. Instead, it makes much more sense to do this in a cell autonomous manner. Okay? So we think that's why we see this kind of picture. Okay, so I just told you that the clock regulates metabolism, but we also know that metabolism regulates the clock. So one example is the deacetylase SIRT1. Deacetylase is both B-mal and PER2 in the core feedback loop. Another nutrient-sensitive kinase, AMPK, regulates the phosphorylation of the cryptochrome proteins, which targets it for ubiquitination and degradation by the proteasome. And so the signals are reciprocal. The clock regulates many metabolic pathways transcriptionally, but then metabolic pathways can also directly impact the core clock system and modulate it. So why is metabolism interesting to us? So one of the reasons we got interested in the importance of timing of metabolism is this very simple experiment done by my colleague back at Northwestern, Fred Turek, where they took mice, and as you know, if you feed mice a high-fat diet, they become obese. It's a model called diet-induced obesity. But instead of having them eat it freely, what they did is restrict the diet to 12 hours each day to either the light interval or the dark interval, okay? And what you see is that the mice that eat during the day in the light, which is the inactive period for a mouse, they gain weight. But the mice that are eating this high-fat diet during the night, during their active period, do not gain weight. And it turned out they had the same activity level, they ate the same amount of food. There's something fundamentally different about how the mouse is metabolizing that diet, depending on whether they eat it in the daytime or the nighttime, okay? So this effect has been, replicated by dozens of labs by today. This is an early replication from Sechin Panda at the Salk Institute, where they also use high-fat diet. So these mice become obese, as you can see in pink here. And then the mice that are restricted to an 8-hour window in the dark period, shown in red, are protected from weight gain, okay? And this kind of effect has been replicated in many different forms. So not just high-fat diet, but Western diet, high-fat, high-carb, and lots of different kinds of protocols, like 5 days on, 2 days off. They're still protected. And now, these are just some early papers in human studies, but now literally there are dozens of human studies showing that time-restricted eating has many beneficial effects on health measures, typically body weight, but sometimes blood pressure, glucose regulation, other things. These are all short-term experiments. Okay, so what I want to focus on really for the rest of my talk is on the effects or the role of the clock on lifespan in mice. And so as all of you know, human lifespan has been increased dramatically, over the last century. But one of the problems is there's still a very significant gap between what we call health span, the time that you're healthy, and the time that you actually die. So in the United States, that gap is almost 10 years on average, very long time, that you might spend the last 10 years of your life not healthy. So we are all interested in trying to solve this problem, to extend health span and make that as close to lifespan as possible, so that you remain healthier longer and you live longer. Okay, so that's one of the goals that many of us have. And it turns out that reducing calories is still today the most effective intervention for extending lifespan. Okay. And so dietary restriction or caloric restriction has many different benefits on many diseases. And it's effective in almost all model organisms where it's been looked at, including primates, non-human primates. But, in humans, this is a very difficult experiment to do. So we only have anecdotal evidence for humans. So when we got interested in this, we looked into the classic mouse experiments, which were done by Richard Weindruck's lab in Wisconsin. And what they showed is as you progressively reduce the caloric intake, you can increase the lifespan significantly out to about 40%. It's a huge increase. Some experiments, they even achieve 50% increase in lifespan. That's very rare. But typical in the field is to reduce calories 30%, and you'll get a 30% increase in lifespan, which is very significant. Imagine if we live 30% longer, right? Okay, so when we looked into these studies, this is their standard protocol. Each mouse was fed 4 feedings per week, 1 daily feeding on Monday, Wednesday mornings, and a double feeding on Friday morning. Okay, so to a circadian person, this is a little bit unusual. You know, mice eat every day. They don't eat every other day and skip weekends. And also we know that the time of eating is also very important for circadian rhythms, and that time feeding can actually alter the phase of metabolic tissues in the body, such as the liver, if you eat at the wrong time. And so we were wondering, you know, what the effect of these prolonged fasting intervals were, and what effect feeding in the daytime versus the nighttime might have on longevity. And so when we got into this, it became clear why they only fed, you know, 3 times a week. It's a lot of work. The experiment lasts 3 to 4 years, depending on how effective your lifespan extension is. And so just to feed every day, 7 days a week, for 3 years is a lot of burden for your staff to achieve this kind of experiment, okay? And to do time restriction, it turned out that would be twice as much work because you have to put on the food and take it off each day. And so no one was going to do that experiment on lifespan, time restricted feeding. And so our solution was really to just develop automation. We already have, you know, a very large automated circadian activity facility. I have more than 2,000 cages that measure running wheel activity under controlled conditions with computerized data collection. And we had about this much space above the cage in these boxes. And so what we decided to do was design a feeding system that we could squeeze into that space. So we wouldn't have to use new or additional space to do the experiment. And so these are feeders that we designed and built ourselves. And initially, we were able to actually make about 500 of these feeders to do the experiment I'm going to tell you about today. Now, there's really no commercial solution for this because you can buy an automated feeding system for a mouse, but it's going to cost you a few thousand to 5,000 a piece. And they don't have computer control of delivery and also sensing when the food is taken. So those are all things that are really not available. Okay, so we use a large pellet. It's about 300 milligrams. And so this is an ad-lib fed mouse on the life cycle, 43 days here. You can see in black the wheel running activity, so it's nocturnal. And the pink dots are when it's taking a food pellet. So this is typical. They eat about 75% of their food at night in their active phase, but they eat a significant fraction during the day when they're resting. Now, mice, you know, do not have consolidated sleep. They have very short bouts of sleep. So they naturally wake up every few hours during their rest phase, and they eat in a groom like this, okay? So when we put a mouse on caloric restriction and we started the food interval each day in the morning at 0, what we found is as the mouse got hungry after a couple of weeks, it eats all the food as soon as it can. Okay, so in 2 hours, it eats all, in this case, 10 or 11 pellets that it's given each day. Okay. So that makes sense. The mouse doesn't know the food's going to be there later. It better eat it when it's there, right? Okay, so that makes a lot of sense. But what that means is when you reduce calories, you're not just reducing calories and maintaining this kind of feeding pattern. You change the entire feeding pattern of the mouse. It goes into extreme time-restricted eating, and it has an extremely long fasting interval of almost 22 hours, okay? And so we know, well, let me say, the same thing happens if you start the feeding interval at the beginning of the night. Eats all the food in 2 hours, okay? And so we know all three parameters, calories, timing of eating, and fasting, are potentially beneficial. What is the relative contribution to both health and lifespan? Okay, so we undertook lifespan experiments. We have the 3 groups I just showed you: ad lib feeding, the ones that eat in 2 hours, either night or day. And then to get around the binge eating, we spread the food out over 12 hours, night or day. And then importantly, we have a 5th group where we spread the food out over 24 hours. So there's no day-night difference, and there's no fasting interval, okay? Here are activity records for individual mice in the 6 groups. They all remain nocturnal. You can see a gradual decline in overall activity as they age. These are 3-year records here in this case. And overlaid in red is their feeding pattern. And so you see they maintain this feeding pattern for their entire life in these 6 groups. And they're very obedient when we spread the food out. That's because they're extremely hungry. So they're waiting for that pellet to come in these cases here. Okay. If we look at their body weight, the ad lib group gradually gains weight until they're about 20 months of age, and then they spontaneously lose it. And then all of the restricted groups maintain low but healthy body weights. And surprising to us, there is no difference in body weight among those 5 different feeding patterns. Okay. So here's the first surprise. I asked my aging colleagues, what do you think of those 3 factors would be the most important? They all said calories. You reduce calories, they live longer, okay? So here is the, what we call the CR spread group. That's the one that eats continuously. Even though they were restricted more than 30%, they only live 10% longer. The mice that eat in the daytime, which is at the wrong time, but they have a fasting interval of either 12 hours or 22 hours, they live another 10% longer for a 20% extension of lifespan. And then finally, the group that eats at the right time, which is at night, they live 35% longer, okay? And surprisingly, there is no difference in median lifespan between the 12-hour fasting and the 22-hour fasting. We think the 2 hours may be slightly better, but it was not significant statistically, okay? This result is extremely surprising to the aging community. The timing of eating makes such a big difference on lifespan. It's the difference between a 10% extension and a 35% extension in lifespan. So what is going on here? So we first asked, are there any phenotypic correlates that actually are associated with longer lifespan? And so if you look at total activity, when the mice are young, there's no association between lifespan and activity level until they reach 18 months of age. And then when they're older, then we see a positive correlation. The ones that are more active do live longer. And this is true essentially across all the groups, where at young ages, there's no significant correlation, but then you can see a positive correlation emerge in some categories later in life. Okay, so activity is sort of a marker for longevity. What about body weight? So I asked my aging colleagues, what is your prediction? They'd say low body weight, longer lifespan, which has been found in previous studies. So we looked at that, and in our ad lib group, there's no correlation at all between body weight and lifespan. And of course, in the calorically restricted groups, there's no correlation because they all have low body weight. So this is a sort of another surprise that body weight is not correlated with lifespan. If we look at some endocrine health measures, glucose, which I'm not showing you, but insulin levels, with age, you see an increase in insulin, leptin, and GLP-1. But all of these are actually rescued by caloric restriction when they're old, especially the groups that live the longest, okay, which are shown here, blue and green. And you can see that the spread group also does not have a very good rescue, okay? So what is the cause of death? So in black 6 mice, cancer is the major cause of death. And so cancer accounts for about 75% of the mortalities in our experiment and in others. And lymphomas and hepatocellular carcinomas are the major cause of death in this strain of mouse, okay? And the liver had the most lesions at autopsy. Interestingly, the mice that live longer do not have a different spectrum of cancers. The only difference is the onset of cancer is delayed, and that correlates with the longer lifespan. Okay, so somehow caloric restriction is just delaying the onset of cancer, and that then becomes the major cause of death, as we see in ad lib mice. Okay. So because the liver had the most lesions, we started with the liver, looked at gene expression there. And if we just compare the ad lib group at 6 versus 19 months of age, okay, we see that 2,000 genes go up and 500 genes go down, okay? And so why did we pick these ages? So 6 is a fully mature mouse, and 19 months is an aged mouse, but at that point, 90% of the ad lib mice are still alive on the survival curve. If you go to 24 months, 30% of them have died already. And so we felt that that time point might be compromised by mice that are on the way out, okay? So that's why we chose this particular time point, 19. Okay, so what are the 2,000 genes that go up? They all have to do with inflammation and immune function. I've never seen a Go analysis come back where it's completely uniform. Almost nothing else shows up. It's amazing. Okay. And then for the genes that go down with age, they all have to do with metabolic pathways. And these are largely under control of the circadian system as well. Okay, so if we convert these changes in gene expression to just percentages and look at all 6 groups, what we see is that the longest lived groups have the smallest change in gene expression, meaning they have the least amount of inflammation and the least decline in metabolic gene expression also. So caloric restriction is rescuing the vast majority of these age-related changes, no matter what form of caloric restriction it is. And so if we look at that more than half of the genes are rescued by any form of caloric restriction. If we add fasting as a parameter, then we see a new category, thermogenesis. And then finally, the longest lived groups reduced to a smaller set of genes. But again, these all have to do with immune responses, both adaptive and innate immune responses. And the perfect gene shown here is interleukin-1 beta, which is directly correlated with lifespan. The longest lived mice have no significant increase in IL-1 beta, and the mice that have the shortest lifespan have the highest levels of IL-1 beta, and likely an indicator of system-wide inflammation. So the other change that happens with age is the amplitude or robustness of circadian gene expression declines. So these are a few cycling genes here. This is BMAL, RL, NR1D1, nuclear receptor, HER1, HER2. In red are the 19-month-old mice, and black is the 6-month-old mice. And so what you see is all of them have lower amplitude except for ARNT-L. And genome-wide, we see that there's a significant decrease in fold change amplitude for all genes that are cycling with no significant change in phase. Okay? And there's a slight reduction in the detection of cycling genes with age, but there's also not much overlap or sharing, so you lose genes that used to be cycling when they're young, and you gain genes that become cycling when they're old. Okay. So if we then look at the extreme group, the calorically restricted mice that eat in 2 hours, what you see is the ones that are eating at night have lots of cycling genes, but the ones that eat in the daytime have only 7 with this very strict cutoff. Okay. And the timing of their phases is shifted 12 hours, shown in yellow. I don't know if you can see that very well. Okay. So one of the things that happens with age, of course, is you do get a reduction in cycling genes, but daytime feeding actually interferes with cycling of many genes, as shown here. The other thing we found is that there are many genes where the level doesn't change, but the waveform is changed by daytime feeding. And so these classic aging genes, such as FOXO3, GEIS2, IGF-1, all have nocturnal patterns under either ad lib or nocturnal feeding, but with daytime feeding, their gene expression patterns are disrupted. Okay, so that's a second effect that we see. So what we've sort of found recently is that if you look at all the classic aging pathways, such as IGF-1 signaling, SIRT1, mTOR signaling, FOXO, AMPK, all of these classic aging and longevity pathways are under circadian control in one metabolic tissue or another, as shown by these purple circles here, okay? And so because with age, the circadian robustness declines and the number of cycling genes goes down, we think that one way to rescue these pathways is either through caloric restriction or some other kind of intervention, okay? And indeed, there are a number of recent studies that have shown that circadian gene networks are actually conserved across many species. So here's a very interesting study from Vera Gorbanova's lab, looking at 26 species that have different lifespans from 3 years to 37 years. And when they looked at the conserved gene expression patterns across all these species in different tissues, 2 modules appeared. One is circadian rhythms, and the other was pluripotency. In a recent human study called the calorie study, which is a 2-year caloric restriction intervention. In this case, they're looking at adipose gene expression. One of the top hits is B-male clock, circadian gene expression. And in this study where they were looking at muscle gene expression by biopsy, circadian clocks are also one of the top hit categories. So we're seeing that clocks are probably playing an important role, not only in mice, but across perhaps many organisms and also perhaps in humans as well. And so what are we doing now? So we're really looking at 3 different sets of questions currently. One is whether time-restricted feeding without enforced caloric restriction can have either health or lifespan benefit. Okay. The second is to test this idea that if we rescue the clock, we can improve health span, lifespan. And we're doing this genetically by using a back transgenic mouse that overexpresses clock about 2-fold globally to see if it can be more healthy and live longer. Okay. And then finally, we're looking for small molecule or transcriptional modulators of clock gene expression to try to boost clock gene expression in another way, ultimately through pharmacology. Okay. And so the time-restricted feeding experiments are being done by Samantha Iams, and we have some initial results on body weight. You can see that body weight is lower with either 8 or 12-hour time-restricted feeding. This is only at night compared to ad lib. And using frailty as a measure of health in mice, we can see that there is actually a benefit of time-restricted feeding on frailty scores, both in males and in females. But males, it was only with 8-hour time-restricted feeding. In females, it was with both 12 and 8-hour. And just overtly, you can clearly see that the mice look younger when you look at them. The clock gene expression experiment is just starting, too early to tell what's going on, but it looks promising that the clock overexpression mice are going to live longer. And so we're really waiting for that result, because that'll be essentially a genetic proof of principle for our idea that if you rescue the clock, you might be able to rescue many pathways involved in aging. And then finally, for small molecule screening, we're really conducting a full court press on this, developing many different kinds of target-based assays so that we can find activators of clock B-male, which is very difficult. It's much easier to find inhibitors, of course, but we already have some hits that show activation, so we think it has the potential to work. So to sort of sum up, our idea really is that the clock is really upstream of essentially all of the hallmark of aging pathways that the field has been studying. And that if we rescue the clock, this might give us a new target that gives you essentially a multi-system rescue of many aging and longevity pathways by targeting just one nodal point in the system. And so I'll stop there. This work was really led by extremely talented postdoc, Vicky Acosta Rodriguez, who's now at the National Institute of Aging. That's a collaboration with Carla Green at UT Southwestern and a number of talented postdocs in the lab who are doing various aspects of this work. Thanks.
You probably knew that I was, sorry, you probably knew that I was gonna ask this. I know that, time restricted feeding had an impact on aging, memory decline, and I'm wondering if you've been age. Sorry. I'm wondering if you have been able to, or you plan to include, memory and cognitive decline as one of the measures, because it'd be really interesting to see how it interacts with the, longevity.
Yeah, so we actually measured context-dependent fear conditioning in these mice at 6 and 19 months of age. But we use the same protocol that we use in our ENU mutagenesis screen, which is an overtraining system, which had 3 pairings over 2 days. And it turned out at 19 months of age, there's no deficit, unfortunately. But I've seen other people using novel object recognition, and they could actually detect something in aged mice. But unfortunately, we didn't do that in this cohort. So I think our fear conditioning paradigm was really not sensitive enough. We should have done 1 pairing or something, Yep.
In the beginning, you emphasized the autonomy of the circadian system in every cell. And the question now with given your results with the calorie restriction or time of day night feeding, is that still consistent with autonomy of cells, or do you want to think about a sort of master organ, such as say the liver, which is sending out signals for time of day feeding?
Yeah, so at the system level, there are many system-wide rhythmic signals that peripheral tissues can respond to, and they're redundant. So there's glucocorticoid, there's melatonin, there's circulating serum factors that act on the serum response element that Uli Schibler discovered, and the subtle circadian body temperature rhythm of a few degrees or 2.5 degrees in a mouse is an extremely strong synchronizing signal for all peripheral tissues in the mouse. So what happens is, in the case of the liver, if you give daytime feeding, the liver flips over, but the brain doesn't. The brain still follows the light, or I should say the SCN, hypothalamus, still remains locked onto the light cycle. And only about, half of the tissues we've looked at in the body actually shift to a nutrient signal. And there are even some tissues where half the genes switch. We haven't done this at the single cell level, but only in bulk RNA-seq. But in skin and in brown fat, when you have daytime feeding, only half the genes follow the feeding signal. The other stay put. And so you get many different kinds of effects like this. But I think one of the important take-home messages is just by altering the time of feeding, you can cause an internal desynchronization at the sort of system level, where the brain and liver are, out of phase. And probably that's why those mice don't live as long as the mice that eat at the right time, where everything is in sync. I don't know if that answers your question or not. Yeah.
Hello. That was a very, very cool talk. So I feel like I've read and there's been like a big emphasis on like just sleep health in general, like I'd like Dr. Walker at Berkeley, and just kind of like shoehorns in a lot of that, or tied in with a lot of what I've been reading about. I know, like, I'm just thinking in terms of like, just a very specific example of like adenosine cycling throughout the day, and how like, for instance, if you like drink caffeine, like block adenosine signaling. My question, I guess, is just like, has there been much research about like how different like behavioral things, whether it's like caffeine or something else, are affecting like the cycling transcription that you talked about, like the circadian transcription cycling? I don't know if that quite made sense, but I was just curious if that had been looked into.
Yeah, I mean, sleep state clearly interacts with the circadian system. So it's a reciprocal relationship. Sleep modulates sort of the, in humans, it's really modulating when we wake up. And when we go to sleep is really dominated by the homeostatic sleep system, which is maybe involving this adenosine-like system that's measuring wakefulness. And so sleep, as far as longevity and aging is concerned, is incredibly important. If you sleep deprive, then you can show that you'll have shorter lifespan. If you desynchronize the circadian system chronically by resetting the light cycle in mice, Jean Block, your chancellor, actually showed many years ago that rats that were phase advanced every week actually died earlier than controlled mice. And mice that were on a delayed light cycle didn't have that effect. So both circadian disruption and also sleep deprivation, have very morbid effects, no question about that.
Do you know if so these circadian, like the genes that are modified by the circadian rhythm, they're expressed different at different times of day? Do those cycles still hold up in vitro systems? And do you know if that's something that is like a potential issue with like long-term experiments in vitro culture, where you might see differential gene expression at different time points based on just like when you sample these cells?
Mm-hmm. Yeah, so, as I showed the fibroblast imaging movie, they have beautiful clocks. But macroscopically, the culture doesn't stay synchronized. You have to give them a synchronizing stimulus, like changing the medium, serum resets them, glucocorticoid, or temperature. Or actually just taking them out of the cell culture incubator and letting them cool off a little bit will reset your cell culture. So probably people don't want to know that, but but it does occur. So if one of the things that is going on at the system level is that there are definitely systemic signals driving gene expression, say in the liver. It's not all autonomous to the liver. It's a combination of the cell autonomous clocks in hepatocytes and other cells, and these system-wide signals that are impinging on the liver. So it's sort of complicated. So in vitro, you can try to isolate that. And depending on the tissue, then you get preservation of many of these cycling cells, genes, I should say. So for example, the suprachiasmatic nucleus actually has many cycling genes, but it's an extremely robust oscillator. If you look at fibroblasts, there are many fewer genes that are cycling when you do an RNA-seq experiment, for example. Even though the per gene is beautiful, you won't see 1,000 genes cycling in a fibroblast. Yeah, Nelson.
Yeah, so, you were saying that the main cause of difference in lifespan is different cancers in the mice.
Mm-hmm.
And I just wondered, are there specific mutations, sort of along the cancer pathway that the different interventions that you're looking at have an impact on affect health?
Yeah, I don't know if I can answer, I don't know the answer to that question. I think we don't really understand why black 6 mice are susceptible to hepatocellular carcinoma, for example.
Mutation pathway towards that.
They're very sensitive to, you know, hepatic steatosis, NASH, and then the progression to HCC. And so probably it's really that sensitivity to diet. So you saw the ad lib mice get, you know, they gain a lot of weight. So they actually have fatty livers when when their body weight is that high.
Thank you so much for the presentation. Wonderful work, and it's great to listen to it.
Thanks.
I've been amazed by the widespread impact, of course, of the circadian rhythm on health and disease and lifespan as you presented. But I'm also surprised, more or less, and I'd like to hear from you if you're surprised too, or maybe I interpreted wrongly, that when you consider the core clock genes, 5 to a dozen maybe, that it seems everything seems to be going back to those handful of genes when it comes to circadian rhythm in terms of its origin and regulation. And that singularity almost of the system is different from what we see in many polygenic traits, neurobehavioral traits, for example. So is it a true observation that it is so confined in those 5 to 12 genes, or is it biologically explainable? Because if you had waken me up today, not knowing anything about circadian rhythm, I would have thought that the drivers of it will be far more than those dozen.
Yeah, I mean, I think, you know, the circadian clock is analogous to the cell cycle, where there's also, you know, what you would call a core set of cell cycle genes. But then they have many transcriptional targets, translation regulations. And so I guess it depends on how you look at the system. So for example, if we started by looking at RNA-seq data without any mutants or clock genes, it would be very puzzling because there are hundreds of thousands of genes that are cycling. But what we know now is most of that is really output that's driven by this core gene network. Now that gene network with the 3 loops is a little more complicated. So maybe it's 30 genes, something like that, but it is still, I think, a pretty finite number.
So, yeah, I was told that we should wrap up.
Okay.
To let Joe have lunch with the students. And so let's thank him